Hello everyone, I will be talking about G-quadruplexes today. DNA is a double helical structure made up of building blocks called nucleotides. The four types of nitrogenous bases are adenine, thymine, guanine, and cytosine. Each nucleotide contains a phosphate group, a pentose sugar, and a nitrogenous base. Of these bases is what determines the DNA's instruction. Human DNA has around 3 billion bases, and more than 99% of those bases are the same in all humans. There are different types of DNA based on their structure. The first kind is the A helix. It is a right-handed double helix structure which is fairly similar to the more common B DNA form, but with a shorter and a more compact helical structure. The bDNA is a right-handed double helix. The double strands of bDNA run in opposite directions. Next is the G helix. It is a left-handed helix. The zigzag-like appearance of the backbone distinguishes gDNA from other forms of DNA. Next is the G-quadruplex. It is formed when guanine-rich DNA sequences fold into four-stranded secondary structures. And lastly, I motif. It is also a four-stranded quadruplex structure formed by cytosine-rich DNA. G-quadruplex, also known as tetraplex or G4 structures, are formed in nucleic acids by sequences that are rich in guanine. The building blocks of G-quadruplexes are G-quartets that are formed through a cyclic Hookestein hydrogen bonding arrangement of four guanines with each other. The planar G quartets stack on top of one another, forming four-stranded helical structures. G-quadruplex formation is driven by monovalent cations such as sodium and potassium, and hence physiological buffer conditions favor tetraplex formation. This is an animation of a tetraplex in a telomere. The tetraplexes are seen at the center of the structure, and it contains three layers of G tetrads. Telomeres are repetitive DNA sequences present at the end of the chromosome that protects the chromosome from degradation. The repeats in the telomeres are TTAZZZ. The three Z's here are responsible for producing the tetraplex. G4 structures are known to help regulate gene expression by activating or downregulating transcription. Transcription is the process by which RNA is synthesized by using DNA as a template. G4 structures can also block DNA replication. DNA replication is the biological process of producing two identical replicas from one original DNA molecule. G4 structures can also control the length of the telomeres in the chromosome. Chromosome lengths are known to play important roles in aging. And finally, G4 structures also regulate how much proteins are being synthesized in the cell. That is the end of my presentation. Thank you for listening.